Hello everyone, and welcome to today's Dr. Ehrenstorfer webinar. Firstly, I'd like to introduce myself. I'm Dominic Keeley, and I am the Global Assistant Brand Manager for Dr. Ehrenstorfer. I'm joined today by Dr. Kelly Cheshire, who is the Global Product Manager at Dr. Ehrenstorfer, and who will be taking you through this really important topic of pharmaceutical and veterinary compounds. Just a little bit of housekeeping before we start. If you do have any questions, please feel free to ask those. To do that, you just type into the control panel box on the right-hand side of your screen. You can do this at any point within the webinar, and we will have a question and answer session towards the end where we'll come to answer those questions. So without further ado, I'll now hand you over to Kelly.
Thanks, Dominic, and hello everyone. Thank you for joining today. As Dominic has already mentioned, my name is Kelly and I'm the Global Product Manager for our environment, food and forensic reference materials, and that includes our fantastic brand, Dr. Ehrenstorfer. I'm excited to be giving today's talk, which will be going through pharmaceutical and veterinary residues in the context of food and environmental safety. And what I'll do is I'll begin with discussing what these compounds actually are, how they get into our food and the environment, and why they should be of concern. And I'll be doing this with particular reference to antibiotic resistance. I will then move on to the regulatory landscape, where I'll discuss what we can do as scientists to ensure the integrity and safety of food and the environment, and how we at LGC are here to support you in this effort. And as Dominic has already mentioned, we will end the talk with a questions and answers section. You can submit these at any point during the talk. If they're not answered during the slides when I'm giving the webinar, we will come to these at the end and we'll do our best to address them. If not, we'll follow up with emails after the webinar today. So without further ado, I will crack on. So pharmaceutical and veterinary medicinal products, what are they? They're basically the products used to treat disease or prevent outbreaks of disease. They can alleviate stress or pain in humans and animals, and in some cases, they're used to improve yield. Now, treating and preventing outbreaks is particularly important for husbandry. So this is the care, cultivation, and breeding of crops and animals. And the reason for this is that husbandry practice involves animals being in close quarters, which means that bacterial and parasitic infections can easily be spread. Now, while the compounds we have to treat these are essential for human and animal welfare, they can leave residues in both the food chain and the environment. And it's important that we look at these compounds and the associated metabolites and how they can impact food and environmental safety, and the consequently negative implications that they can have on human, animal, and environmental health. So there are various ways in which veterinary medicinal products can enter our food. So that includes the purposeful modification to treat diseases. So these can be administered to individuals via injection. They can be topical, oral, or through implant. For flocks, herds, or ponds, these compounds will likely be added to feed or as water additive or a pour-on or drench. We also have cases where it's possible for healthy animals to eat contaminated feed, or even after an animal has been taken off a medication, they might still be able to get hold of contaminated feed, which would then affect the withdrawal periods and consequently end up in the food product at the end. And the withdrawal period is is basically the time taken from when the medicinal compound is finished being administered to the animal to treat the disease to the point of when the food product is collected, so when the animal is slaughtered or the crop cultivated. And when these compounds enter the body, they break down by a process known as metabolism. But if they're not fully broken down before they are turned into a food product, so things like meat, dairy, eggs, so on, they can leave residues behind which are then consumed by humans, impacting their health. Now, this may be the case, as I mentioned, if the incorrect withdrawal period is followed or if the incorrect dosing is administered to the animal prior to collecting the food product. So we need to consider the safety of these compounds and the metabolites when we are using them. Each of the VMPs have a marketing authorization, and this basically sets out what the specific use of the compound should be in each country of sale. Now, all medicines, whether they're human or for veterinary use, can only be sold if they have this marketing authorization in the country of sale. And part of that assessment includes the manufacturer providing evidence from toxicological studies and conservative safety factors of the residue concentration that will have no impact on consumers. And also it means that the time interval from the treatment of the animal and the slaughter is allowed to carry out so that they deplete to this safe level. The dosing level and withdrawal period are dictated for each species within this market authorization, and the associated residue is then set out as a maximum residue limit in law. And I'll be discussing this later when I go through the regulations themselves. But basically, as long as the withdrawal period is followed and the approved dosage is used, the residue should then deplete to the safe maximum residue limit. We then need to consider also how these compounds get into the environment. So we have the manufacture of the products themselves. We have the waste from this, so industrial effluent and spills. These can then enter the earth, into our water systems and so forth. We also have the consumption of these compounds. So there's human consumption, obviously. We excrete them ourselves in our bodily fluids and waste. We also have the farming processes themselves. So obviously animals consume these compounds and will excrete them. We also have manure or slurry spread on farmland. And again, this can then go into the crops, which are then again consumed by humans. And then that enters us. We also have waste products. So imagine that you have been prescribed a course of medication. Maybe you don't finish that medication because you suddenly feel better and you don't think you need to take it anymore. Now maybe you leave this lying in a drawer for a while and then suddenly it's expired. I'm now going to put this into the rubbish, and then it ends up in a landfill site and consequently again ends up in the earth, ends up in our water supplies and so on. Now when these residues in the ground or surface or sewage water then go through to our water treatment facilities, and obviously this cleans up the water to make it safe for us to then use. But that doesn't necessarily mean it captures everything, and sometimes there can be residues that are left behind in our drinking water, which again we consume and then can leave residues in ourselves. But also we would then use this water for things like cleaning and gardening, and again we're spreading it back out into the environment. And again, as part of that market authorization for these compounds, you have to do an environmental risk assessment as part of this to ensure the safety of residues within the environment. And the kind of residues we're talking about here are things like non-steroidal anti-inflammatory drugs. We've got anticonvulsants, opioids, hormones, and antibiotics. And again, depending on which group of residues are left behind can then vary the impact this has on consumer health. So some of these compounds left behind might be carcinogenic. Some can cause renal failure in birds. Hormones can interfere with reproduction. And then if there are antibiotics behind, this can give rise to something called antibiotic resistance, which is what I'm going to move on to now. So what is antibiotic resistance? It's the adaptive change of bacteria, otherwise known as a mutation. And basically this means that it allows them to grow in the presence of a drug, which would normally either slow their growth or kill them. And what this means is if we've got antibiotic resistant bacteria or fungi, they become harder and harder to treat, and it can lead to increased morbidity. So if we take some antibiotics and we give it to livestock to keep them healthy, either to treat an infection that's already there or to prevent an outbreak. These antibiotics protect the livestock from known forms of bacterial infection. However, some bacteria can then start to resist that drug and mutate. This mutated form of bacteria then contaminates the meat product, and which we can then consume as humans, which then passes on this resistant strain of bacteria. And then when the same antibiotics are given to a human to treat this disease, it may have no impact or have a limited impact because the pathogen is becoming immune. And in 2019, there was a survey of the EU member states by the European Food Safety Authority. And what this found was that the number one food safety concern was the misuse of antibiotics, hormones, and steroids in farm animals. And the reason this is of such great concern is because it gives rise to this phenomenon, and infections, things such as like pneumonia and tuberculosis, become harder and harder to treat. And therefore, the widespread use of antibiotics in medicinal and agricultural practices is only accelerating that process. This is gaining more and more traction in the news and raising public concern. And what I've done is picked out some articles for you today that actually highlight the urgency of this issue. So it's been noted in a 2016 report by the UK government that around 700,000 people die annually due to antibiotic resistance. And studies have shown that this is only set to increase. So by 2050, they estimate there'll be 10 million people a year that die due to this phenomenon, which is just incredible. In other studies, it's shown that it's an even more urgent prospect than things such as pandemics. So as we can all imagine right now with the coronavirus, we know the effect and the devastation that this has had on the population, to our health, to the day-to-day running of our lives. And you consider that this phenomenon is even more critical than that. It's a really, really scary prospect. And the reason it's considered slightly more urgent is that some of our most vital medicines have just simply ceased to work or to be effective against disease that we use. So things like hip operations and cesareans, even that could become fatal. Even things like UTIs or sexually transmitted diseases, which are easily treated today, could become fatal as well, which is, as I say, a very scary prospect to consider. And this is only exacerbated further by the overuse of veterinary medicinal products in agriculture. So the UN is actually seeking an agreement for a global action plan for how we go about finding new antibiotics and diagnostic tools and how we can prevent this phenomenon from occurring. And while new antibiotics would be great, this is only a short-term fix. In the long term, what we need to think about are system changes. So things such as clean water being available, making sure that appropriate hygiene practices are in place, and that we are responsibly using these compounds in industry. And part of that is obviously making sure that we test for them thoroughly with regulations, which I'll come on to in a second. With regards to hygiene practices, this is not just in industries, but we should also consider it for our own day-to-day life as well. So I even came across an article where because of these compounds and their metabolites being excreted in our bodily fluids, so in our sweat, we go into the shower to wash off an illness, and all these compounds are then coming out in our sweat and being washed down the drain. Now, if you think of the drain, it's this dark, moist, kind of warm environment, which is like the perfect breeding ground for this bacteria. And once again, this can then enter into our water systems, so go into water treatment facilities, and again becomes wider spread in the environment, just increasing that problem. And speaking as a pet lover myself, I know the UK is a nation of pet lovers, but I even came across a study where resistant bacteria is actually being passed on from pets to their owners because of these kind of compounds being left behind in pet food, which is something you don't generally think about on a day-to-day basis. So it's really, really important that we have responsible use of these and that it is regulated to ensure that this applies. So thankfully, we do have some, we have regulatory bodies now that are tackling this issue. So we have the EFSA, we have the EEA and the EPA, which are looking at antibiotic resistance in food-producing animals and in the environment. But not only this, we have a lot of other areas where we need to regulate compounds. So there are more considerations. Obviously, antibiotics I've highlighted here because we've covered that, but we also have things like non-steroidal anti-inflammatory drugs. We have antimicrobials, antifungals, sedatives, so many more that we need to regulate. And we need to screen for these in our products. So that brings us on to regulations. What are regulations? Well, they're there basically to ensure compliance in dosing and withdrawal periods with regards to food safety and the appropriate disposal for environmental safety. And this is in alignment with the criteria in the marketing authorization of each compound. And the regulations set out this maximum residue limit value, which I alluded to earlier. And this is for each compound and metabolite with regards to food testing, and it's also specific to species, so cattle, fish, sheep, chicken, and it's also specific to the type of tissue that we get from these animals, so kidney or muscle, or even other food of animal origin, so milk, eggs, and honey, for example. The environmental testing side of things is typically associated with water and soil quality. And these maximum residue levels are set by working backwards from the acceptable daily intake level. So this is basically what the safety limit is for using that compound, which is slightly different to pesticides. And for the lab analysis of these kind of samples, generally you would do an initial screening, and this eliminates any negative samples. And typically you would look at half the compliance limit as a trigger for confirmation of this. Liquid chromatography coupled to a mass spectrometer is used, and this requires an RM of each drug each day spiked at half of that limit. Anything that comes through as a positive screen are then retested to confirm and quantify those compounds, and you would use a more representative subsampling and replicate subsamples as needed. Again, LCMS would typically be used for that, and you would use a reference standard and an isotopic internal standard for the drug of interest. The reason we use this isotopic internal standard is it can improve the accuracy and precision of your analyses and enables reliable recovery, correction, and compensates for matrix effects. And this is because hyphenated mass spec methods are highly fractionating, and this can vary over a period of an analysis. And therefore, measuring a material of known isotopic composition and fractionation, that means it can be taken into account in the data analysis and interpretation that follows. We also have prohibited substances. So for some veterinary medicinal products, there's no safe residue limits that have been agreed upon. And this can be because of toxicological concerns or because data submissions for historical approval have been incomplete by today's standards. And these are prohibited for use in food-producing animals. So that would include things like nitrofurans, phenylbutazone, and chlorpromazine. The EU also prohibits the use of steroids, hormones, growth promoters, or antibiotics where the main function is to aid feed conversion as opposed to treating a disease. And the EU has a zero-tolerance approach to these residues. And in these cases, we have what's called a reference point for action value. And that's determined by toxicological risk assessment, and they're used as a benchmark to ensure the EU guidelines are being adhered to. And why is this important? Basically, because of this, the relevance of an increasingly globalization means that we need to show agreement between different regions and different territories. So just because something is prohibited in the EU doesn't necessarily mean it's prohibited elsewhere in the world. But if you are trading with the EU, you would need to prove your compliance with their rules. So for instance, if a product was coming from the US into the US must provide evidence of the measures being taken to meet the levels set, and the values arrived at must be accepted by the EU. So what does this mean? Basically, in this scenario, imports from the countries using these compounds must demonstrate what's called a split production system. So basically, they would need to have a dedicated slaughterhouse that's licensed by the EU, and then only takes animals that have been raised in accordance with these EU regulations. And some of these regulations are listed on the next slide. So we have EC 9623. This is an EU directive that sets out requirements for residue monitoring plans and establishes the range of substances that need to be tested in live animals and animal products. And within that, each EU member state must have a national residue control plan. And these are targeted surveillance testing designed to police the correct use of these medicinal products. And non-EU countries exporting food to the EU must implement the same kind of monitoring programs and comply with the equivalent food safety levels. We also have EU 2010-37. It's another main one for the EU, and it contains the pharmacologically active substances and their classification regarding to maximum residue limits in foodstuffs of animal origin. And then a more recent addition to this is actually EU 29/1871, and this goes over the reference point for actions for nitrofurans and its associated metabolites. In the US, we have EPA method 1964, and again, this looks at the monitoring plans and the pharmaceutical residues in water, soil, and in sediment. For China, one of the main methods is GB 31650 2019. And again, this goes over the maximum residue limits for a number of veterinary residues the National Food Safety Standard. It's something like 260 analytes within there. So as I mentioned before, there's a newer regulation which sets out the RPAs for nitrofurans. Now, nitrofurans are a group of antibiotics or antimicrobials that are generally prohibited in the EU because they can be used as a feed conversion. But obviously, even though they're prohibited, we still need to test that these products, that these residues aren't left behind to ensure that somebody hasn't been using them. So it's compliant with the regulation that's being tested for. And I've just highlighted here some of the main nitrofurans and the associated metabolite that they're suggesting you test for and the recommended reference point for action. And we actually have a number of these in the Dr. Ehrenstorfer portfolio to help support in the testing of this method. And why is that important? Well, it's important that we test for metabolites. As the analysis of metabolites ensures we've got compliance with regulations and can prove illegal use. Now, metabolites are formed by natural biochemical processes in humans, insects, animals, plants, and microorganisms. That's to inactivate and excrete an unwanted chemical. Now, parent compounds that sometimes metabolize fully in just a few hours or days, and therefore abuse would not be detected if checking only for the parent itself. The maximum residue levels that are derived from safety limits, their main purpose is for regulatory bodies to monitor and enforce adherence to those dosing authorizations and withdrawal periods. So we have to include these metabolites because checking for them could reveal abuse. So what I've done here is taken one of the nitrofurans that's in this latest update, and you can see it's broken down into a number of different metabolites. Now, nitrofurans metabolize very, very quickly in certain species. So things like chickens, they can break down in a matter of hours. So if you were looking just for the parent compound, you might miss it and not be able to prove that it's been used. Now, in the 1990s, nitrofurans were banned from being administered to food-producing animals in most jurisdictions. And for a few years, no issues were detected. But as test methods developed and became more sensitive, we were able to test for tissue-bound metabolites. And what was actually found was that there are a number of nitrofurans still in use in the industry. And we found that there are residues in meat and fish that can only be detected as metabolites. Another example is veterinary antibiotic carbadox. So this is used as a feed additive to promote weight gain in pigs. And although carbadox itself can have genotoxic and tetragenic effects, it is completely metabolized. And the main metabolite, QCA, was considered safe. Again, in the 1990s, the World Health Organization and the UN Joint Evaluation Committee for Food Additives concluded that there were no residue concerns with carbadox used in pigs. And on this basis, carbadox still was permitted for use in some areas around the world, although in other jurisdictions such as Canada and the EU, it was only allowed to be permitted if not used as a growth promoter. Now, there are two other intermediate metabolites that are created, and one of those is desoxycarbadox. And what was found with more modern test techniques is that traces of desoxycarbadox actually outlasted QCA in edible pig tissues. And the reason this was of concern is desoxycarbadox was actually suspected of being a more potent mutagen than carbadox itself. And therefore, the FDA served notice to ban that, along with other jurisdictions, provided a manufacturer cannot provide safety evidence. So an example of when we would test for these metabolites, we would test for QCA if we're trying to police a carbadox ban and ensure compliance with a regulation. But we also might test for desoxycarbadox as well to check for exposure studies or for toxicity studies. So how can Dr. Ehrenstorfer support you in testing for these compounds? So in the last year and a half or so, we've been doing significant MPD in this area, and we've increased our portfolio by 25%. We now have over 1,000 reference materials in this portfolio, and it includes metabolites and stable isotope labeled compounds. We also have multiple formats available for you. So there are neats, we have single solutions, and we even have some regulatory mixtures to make that analysis more streamlined for you. We also have a significant proportion of our portfolio available as ISO 17034 accreditation. And all of these compounds can be found in our new pharmaceutical and veterinary compounds brochure, which is available on our website. Our website has also undergone some significant changes over the last few months to improve your search experience. So we have regulatory method filters, analyte filters, and this should help you find your products more easily. We also have more clear categorization on there, including labeling of metabolites. And on significant interest to you be on parent product pages, we also have our related products at the bottom of the pages now. So you can find the metabolites and stable isotope labeled compounds that you would need to facilitate your analysis and ensure safe testing. So to conclude today's talk, it's necessary to test for pharmaceutical and veterinary medicinal residues in both the food and environment to ensure safe use and safe practice. The criteria for which are set out in regulations worldwide, and as a food producer, you must provide proof of adherence to local regulations. Not only that, you must also prove safe waste disposal practices as well to ensure environmental safety. And it's also important to test not only the parent compounds that are being used and administered, we also must check for the metabolites as well, as these parent compounds can be metabolized incredibly quickly. And on that note, we are on to the questions and answer section.
Thank you very much, Kelly. Just whilst you have a moment to kind of pop your questions into the box, I just want to make you aware of further reading around this topic. So there are a number of Dr. Ehrenstorfer white papers available. The first one to take a look at is Residues of Veterinary Medicines in Food, and that's looking at the regulation and testing within the EU. That does go into more depth in terms of testing methods, so I'd definitely check that out if you've not already done so. We also have a second white paper, which is along the same lines, but looks at regulation and testing outside of the EU. And the third white paper is Residues of Pharmaceuticals in the Environment. These are all available on the Dr. Ehrenstorfer website or by Googling them. Just another mention as well, if you're not already connected with LGC and Dr. Ehrenstorfer, please do so on our social media platforms, both on LinkedIn at LGC Standards and Dr. Ehrenstorfer, and also on Twitter at LGC Standards and at Dr. Ehrenstorfer. We post regularly on there and keep you up to date with the latest news, white papers and information around these topics. Also, if you do have any questions that you might come to later on, please feel free to email Kelly directly at kelly.cheshire@lgcgroup.com. So we'll just give you a few moments to pop any questions that you may have into the box. So just the first question is asking about slides for the presentation. They will be available. You will receive an email tomorrow, which will contain both a recording to this webinar, so you can rewatch it, and also a PDF to the slides, so again, you can view those in your own time. So the first question we have is, in what concentration range are pharmaceutical and veterinary compounds being found in the environment?
Generally speaking, it's in the nanogram per litre to microgram per litre kind of range, but for exact values, I don't have those to hand, I'm afraid. I can consult our technical team, though. They might have more information on that.
Brilliant. Thank you, Kelly. Another question we've had in is, are Pharma vet res metabolites hard to synthesize?
Again, our technical team would be more suitable to answer that, but from what I understand, compared to something like pesticide metabolites, they're relatively straightforward to synthesize. Generally speaking, I think it's in around 2 to 4 steps. Obviously, there'll be deviations from that as well.
Brilliant. Thank you, Kelly.
Understand that.
So we've had a few questions which will require a little bit more kind of technical input. So we will come back to you on these via email just to ensure that we give you the correct information. We'll give it a few more moments just in case anyone else does have any questions before we conclude the webinar. Okay, it doesn't seem we have any further questions. So all that's left to do is say thank you very much for joining us today. I hope you enjoyed the webinar. As I said, if you do have any questions that come to you later on, please feel free to email them directly to Kelly or your local sales representative. And that information can be found on our website. But thank you very much once again, and I hope to see you again soon.
Thank you, everyone.